Can you see my slides okay?
Yes, we can. Oh, so what are the in this picture, what are the tides like there?
There's a 13-foot tidal range in my study area on the outer coast, which feels a bit small, actually. We've got a 25-foot tidal range in Juneau, where I live. And then other parts of Alaska have even more extreme tides, like the Cook Inlet can have up to 50-foot tidal ranges.
Yeah, I've been there and saw that. That is amazing
It's
how much
huge.
water can go, you know, and for somebody from Wisconsin, that's just a totally foreign concept. Like, where does all the water go?
Definitely. And it's something you totally have to think about as a boater. Jeff, do you want a few more people to hop on, or should I just take it away?
Where are we at here?
6:30.
All right. Anybody in the queue? What are we at?
Nope. We got 26 people on.
Okay, well, Jenell, why don't you take it away and um just a small quick introduction. And I don't remember if it was your mom that suggested it or somebody from the Waupun community had suggested like we got talking about birds and like, oh, we know somebody that, you know, studies shorebird and she's local. And like, oh, this is just great. And um just a little bit, I want to, you know, tell us about your journey, how you went from Waupun and ended up in Alaska and the whole thing. So inspire us.
All right, I'm happy to. It's good to see you all tonight. I definitely believe it was my mom who said, "Hey, my daughter would be happy to give a talk." And I am. I grew up in Waupun and spent time in the Marsh as a kid. I would not say that I dreamt of doing ornithology as a profession, although I always enjoyed birds, being around them. My family did a lot of waterfowl hunting growing up, and in the spring, turkey hunting. And that was always a fun time just to watch birds. Like, I have fond memories of sitting there in the late afternoon and seeing tanagers and snipe and those cool aerial displays that they do. So I've always been interested in birds, but I never considered it to be something that I was going to be doing as part of my career. I was really interested in marine science from a young age, even though growing up in Wisconsin and growing up landlocked, I didn't have a ton of ocean experience. I just loved being in and around the water. And when I was in high school, I volunteered for the DNR, and I spent some time doing everything from invasive species removal. So teasel, that plant that has the really pokey, spiny tufts. I did a lot of removal of that. And then I participated in some bird banding activities. We were trapping mourning doves. We weren't very successful, so I didn't get a ton of banding experience doing that. And then I also did things like go out and do vegetation surveys and got to ride around in the DNR airboat and go out and do surveys. So that was really inspiring and fun. And I just kind of continued on that line of being interested in biology, but knew I wanted to spend some time at the ocean. And so I went and pursued an undergraduate degree in marine science. And I went to Jacksonville University in Florida. And that was great. I love Florida. I spent time with doing independent studies, studying bottlenose dolphins in estuary environments, and landed a few internships that brought me to both Maine and Alaska. And I loved both of those states, and ended up pursuing a graduate degree at the University of Alaska Fairbanks and studied walrus for my PhD. And then upon finishing that, I applied to a job at the Alaska Department of Fish and Game as their endangered species biologist. And they were looking for somebody who had a lot of different wildlife background. And so in addition to doing that sort of stuff, I had also assisted a woman doing her PhD on large whale distribution. I had done that internship in Seward that I talked to you about, Jeff, with stellar sea lions, and I worked on fisheries. I was a technician and a lot of salmon research in the summers, putting myself through graduate school, and even participated in studies on moose and caribou hormones. And so they ended up thinking I was a really great candidate for this job because I had a plethora of experience. And what I do is a little bit of a blend of policy and research. The research part is what's really fun, and that's what gets me out into the field every year. And so that's what I'll talk to you about today. So I've titled this talk Counting Red Knots for Conservation. I'll share with you a little bit about why we're studying red knots. I'll tell you about this species and its life history. I am studying the Rosalari, or Pacific subspecies of red knot. I know you guys in the Horicon Marsh have seen red knots before. Probably they belong to the Rufa or Atlantic subspecies. I'll chat with you about the big goal of this project, and then of course, share some of the exciting results after having 2 seasons in Controller Bay, Alaska. So why study red knots and shorebirds in general? Folks that are aware of this big information probably know that we see some declining numbers in different bird taxa across the US. A big publication came out in 2019 in the Journal of Science, which documented about a 30% loss of birds in North America since the 1970s. And so for all bird taxa, this wasn't bad news. You can see here that waterfowl actually are having an increase in recent years, but we are pretty concerned about shorebirds, and you'll see that we've lost nearly 40% of shorebirds since that time. For picking out my study, I started this job in 2020 and started developing a project and kind of was given these boundaries. You know, I work in a non-game program, so I was going to be studying probably birds or amphibians, maybe small mammals. And then we use various ranking systems in which we track how populations are doing specifically in Alaska. And so we use what's called the Alaska Species Ranking System for this. And that's that table in red. And one of the things that really caught my eye was how many bird species were really hitting that list, and particularly how many shorebird species. And so thinking about that, many of you are probably familiar with other watch lists like the one that the Audubon puts out. And then also more Alaska specific, I investigated the Alaska Shorebird Conservation Plan, which the Alaska Shorebird Group puts together, and I'm an executive committee member on that group now. But when I was just getting started, I really was diving into the weeds to figure out what I wanted to work on. I mentioned I really love being around marsh and the water, and so studying a coastal species like shorebirds was much more attractive to me than the idea of sitting in a blind at 60 below trying to catch snowy owls in the North Slope. So I ruled that one out pretty quick. But I also wanted to study a species where we didn't know much about it and there hadn't been a lot of conservation effort to it already. And so the bar-tailed godwits have had a good deal of research going on in Alaska, and the buff-breasted sandpiper actually has too. So that really led me to study red knots. There's only one other project that's been going on in the state for red knots in recent years. So it was clear that we needed to know more about them. They're considered a declining shorebird species, and so they're on our watch list. So these are a cosmopolitan type of sandpiper. There are 6 recognized subspecies of red knots, and the Rosalari population actually has the smallest abundance. So this table is outdated. Our most recent estimate is about 22,000 on the Pacific coast. And we start to get concerned when we see shorebird populations numbering in the 20,000s. That's typically kind of a red light going off that says there might be something going on here with this particular species. And so you can see it's actually the lowest here from all of those around the globe that we know about. So what about red knots in general? Like, how are they doing across the different continents, and what are some of the threats that we see to this species? In general, they're trending downward, and some of the threats that we see are climate change, decreasing prey abundance, and invasive species. So I just wanted to provide for you a few examples about what we do know for red knots. And so one study that was done with the European subspecies found that earlier snowmelts have been correlated with shrinking body size and bill length. And this is really important because it can prohibit their ability to access really high quality prey. Red knots are known as being bivalve specialists. So bivalves being clams. These are really great prey items because they contain really fatty, lipid-rich material. And that's important for red knots because they bulk up and then they burn through those storage, fat storages when they're in flight. So if your bill isn't long enough to get down to those, some of those birds in this study they found out were actually eating plant rhizomes, which are not going to be very good fuel. Another study was here on the US coast. There's actually been a good deal of research gone on. Many of you might be familiar with red knots on the east coast of the US. They're famous for eating horseshoe crab eggs. And about 90% of that population can be documented in Delaware Bay, foraging on horseshoe crabs' eggs. And so in the early 2000s, they noticed that there were some population declines, and this was really due to an unmanaged horseshoe crab fishery. And at the time, the horseshoe crabs were being killed for bait in an American eel fishery and even a conch fishery in the mid-Atlantic. So what's going on in the Pacific? We don't really know, but one hypothesis could be due to an invasive species known as smooth cordwood or Spartina. And so at a really important stopover site for Pacific red knots in Willapa Bay in Washington state, they noticed that this Spartina plant really took over. And what happens is these plants trap the sediment and create hypoxic conditions, which means it's a really difficult habitat for bivalves to live in. And so because of that loss of prey, it's potentially possible that this could have triggered a little bit of a decline for our Pacific population. We do think that the majority of the Pacific red knots stop in that particular bay. So since that time, Washington State's done a lot of work to clean up the Spartina, and we are hopeful that that will be helpful for red knots. So where are they breeding? This species or subspecies breeds in Alaska and in Russia. The part of the state that they're breeding in Alaska is the western and the northern parts of the state, and they actually breed on the tundra. So they go from being this coastal species feeding on potentially bivalves or even fish eggs to living on the tundra and feeding on things like arthropods, so insects. To orient you to my study area, this is a stopover study. So I'm really looking at one area that they stop and forage and fuel up before they head up to their breeding grounds. And this is on the outer coast. The area is called Controller Bay. It's 70 miles south of the nearest town, which is Cordova. And it's a really wild, remote area. It's got a maritime climate that's wet. And I brought up this chart picture. I really wanted to show you that this bay is very shallow. It's totally an expansive, open mudflat, 13-foot tidal range. So on the big days, you're having a 13-foot tide, but that's only happening about once a month. Most of the time, you're seeing 5, 6, 7, 8-foot tides, and it's super shallow. I mean, when the tide is out, you can walk for miles and miles. You could probably walk 10, even maybe 20 miles to some of these outer islands. So I didn't just pick this place because it was an interesting area to study. There was actually another researcher from the Cordova area that put telemetry tags on these birds in Washington state and then followed them up and wanted to know where they were stopping. So it was already well known that the Copper River Delta supplies great habitat for many shorebird species, but we didn't know where else they were going. And so they flew up the coast, and using telemetry, they found out that many of the birds in their sample size were stopping in Controller Bay. And previously this had been undocumented and unknown. And so this bay became really of importance to look at. Not only is the bay important to shorebirds, but it's also important to people. It has a really long history of resource extraction in the state. Controller Bay was actually Alaska's very first oil field. So at the turn of the century, some small settlements and a boom town called Katala all popped up. And they even harvested some coal out of the Bering River. And so the Bering River, which I'll show you later, is part of my study area. And there's the Bering Glacier in that area. And so it was an area that had a lot going on, but then pretty much abruptly after the 1920s, people abandoned it, and there hasn't been an established settlement since. However, there continues to be some interest for resource extraction. So in 2019, Cassandra Energy requested a permit for oil and gas exploration, but there was really strong opposition from the fishing community, from local Cordova residents, from the Alaska Native EAC Preservation Council, and really that was about protecting salmon habitat. So folks know that this watershed is really important for rearing salmon, and most people living in this part of the state make their livelihood on salmon fishing. So not only is it important to the birds, but it's important to the fish. And we think that likely in the future, Controller Bay is going to continue to be probed for resource extraction. So if this does happen and there's some sort of development, even something like logging in this area, they would have to take in a barge, dredge out the bay, which of course would disturb that sediment and many of the bivalves that shorebirds are eating. And so it's important for us to have some baseline data on the abundance of shorebirds, on the importance of areas that they're using, where they're roosting, and what they're feeding on. So that way we can provide people with the information that they need if this area ever gets developed. So that's all the big background information of why I study this area. Well, because it could be considered critical habitat in a biological sense. And so building on the work that other people have done, I was really interested in determining the number of red knots that are using the stopover location and also their diet, figuring out what attracts them to be here and what is it that they're relying on to fuel them to fly further north. To do this, I'm using an on-the-ground approach using mark resite methods. And so this approach really relies on having a marked animal that can be identified to the individual level. And to do this, for many bird species, they use leg flags. And so you can see on this red knot, it's got a light green flag with black alphanumerics, so a combination of letters and numbers, and that is unique to the bird. The color is specific to where the bird's tagged. So people that are tagging birds in North America should be using green. People in Mexico usually yellow. It could be red if you're in Chile. It's different countries have different colors, but then that alphanumeric code is really unique to that individual bird. So they're not going to send out another duplicate of 6HY. My study is doing this mark resite approach to estimate abundance, so the total population in an area, but it's also a really useful approach for people that want to look at connectivity, looking at the total range and life cycle of birds across the globe. And if you get data on individuals across time, you can actually estimate their survival. So as an undergrad, when I would hear about people doing these mark resite approaches, I was always really curious how that worked. And so I just wanted to provide kind of a backbone for what that looks like. How do we get abundance just by having these marked individuals? We use a pretty complex model in order to do that. And I work with statisticians in order to create these estimates that are robust. But just the general idea for estimating abundance, you have to have a number of individually marked birds that's known. And then also you need to know the proportion of birds that are marked in your study population. And so this is just an example. So for my study, say we're on Controller Bay for 3 weeks. We've got observers out there that are conducting what's called scans, and they find 37 individuals across those 3 weeks. We've also got people out there that are doing surveys, and they're looking at the proportion of banded birds versus those that are unbanded. And from that survey data, let's say we know it's 1%. You can do that math and find out that you have 3,700 red knots in your study area. So that's just kind of the basic premise. So I first started this in 2022, and I was able to put together a pilot season. This was a really big project because no other group has done any type of survey in this bay for any type of wildlife or fish species at all. They've done aerial surveys for moose and some big game, but nothing boots on the ground, not even for salmon research. And so it was a big undertaking. I've had a lot of help from partners that are at their local Fish and Game office and local Forest Service office. We managed 3 field camps across the bay, and I had 15 people participate at all various levels. And our goal was to really survey the entire bay and figure out where these birds were, how we could access them. And so we used fat tire bikes. We walked around in waders. One camp had a skiff. We had some inflatable pack rafts to get around. It really was just ground truthing if this approach was going to work. And it's a big area. So this is showing you the bay. It's broken up into 4 different survey polygons, each one being a different color. And I designed this to be about 5 miles for each one. So it would be like 10 miles down and back from any of these polygons. However, I can tell you that nothing is as the crow flies when you're actually doing these surveys. So that white polygon where we surveyed in 2022, I stayed with that blue tent is approximately, if you wanted to go all the way down and back from camp, that would be a 20-mile day walking on a mud flat and doing multiple stream crossings. So nothing is fast when you're out there. So before we get to the great, exciting results, I really want to share some of the challenges of developing a project like this. I'm not going to go through everyone on this list, but I do have some fun photos to show you guys of what it was like our first season out there. So our first season, we didn't time the migration super well. And the first week it stormed like crazy. We had 45 to 60 mile an hour winds. And the last night it blew 80 miles an hour and shredded our tent. Another challenge we had was just something called chill blains. So if you're working in the Arctic or coastal Alaska where your hands are damp and cold, they can break out in blisters. Just figuring out how to get across the bay. It's really muddy water, so you don't know how deep some of these little streams are. And so you could walk in and be 6 feet underwater over your waders. And so we had to bring out pack rafts and figure out, oh, should we leave the bike, or how do we safely get the bike across and keep going? And those were some real challenges. We didn't really even know if the bikes would work. Some people that had been to this area said, oh, bikes would work great. Other people said, oh, it's like really, really slick mud. You can't bike there. We found both those things to be true. And so in the picture on the upper left is my colleague Charlotte on sandy terrain that's really good for biking. And then the picture on the right is my technician Lyda, who was biking in a really greasy part of the bay that was really challenging to stay on your bike for. Another challenge we had at one of our camps was brown bears. This area has a lot of different predators. There's brown bears, black bears, wolves, and coyotes. So it's a really unique ecosystem, even by Alaska's standards. We saw all of these things while we were out there. And so the brown bears, as soon as that weather turned from rainy and really crappy to getting nice and cold and crisp, came out in full force. And so they like to cruise that bay. It's shallow mud flat. It's an opportunity for them to go clamming and looking for marine mammal carcasses like whales and sea lions that might wash up dead on the beach for them to eat. But then also May is their breeding season. So we had a lot of what we called bear TV in the evenings where we'd pull out some lawn chairs and watch what was going on. And there was this particular female with 2 cubs in the lower left picture, and this large boar that was just pursuing her to breed with her. And eventually, the very last day we were out there, she booted her 2 cubs, presumably to breed with this male. So although these were fun interactions to see, it was not great to have a bunch of bears around your field camp. We had to be super, super on it with not having food out, with having the bear fences running, with having firearms and that sort of stuff. And then it was really hard to do the work because every time it was low tide and we wanted to go look for these red knots with leg flags, there would be bears in the mud flat. And so we just couldn't go out often. And then weather, everybody knows that weather is unpredictable. And in this part of coastal Alaska, one of the main issues is fog banks. So when the air is cool and the ocean is warm, it can create these conditions where fog just rolls in and completely socks the area in for days at a time. And it can create these pea soup conditions, which make it difficult for your optics to focus on birds. And then this one, our pilot got our plane stuck, his plane stuck. And that was not a very fun time either. He was pretty stressed out because it was an incoming tide. So the crew was able to dig him out, but we had a lot of things to figure out in 2022. Overall, this was our pilot year. We didn't have enough marked individuals recorded in order to make this model work and get an abundance estimate. Technicians really don't like to hear that, but it's not like this year was useless for us. It was really useful. We were able to figure out how to time the migration better. We cut out one of those field camps, the one that had all the bears and none of the red knots. We just completely cut that one out and decided not to survey there. And we figured out where these birds were roosting. And then I also hired dedicated photographers. So that first season, people just went out with spotting scopes and binoculars to record those leg flags, but we ended up having a bunch of leg flags that either didn't match any current flags in the system. So it was hard to say, oh, was this observer error? Or did someone write down the correct flag, but it just wasn't captured, so we have no record of it. So these were some really big adjustments we made in 2023, and it was super helpful to us because we had a very successful season. So from 2023, I just wanted to show you guys a few of these money shots that our photographers took. So this one, the red knots are foraging. You can see little raindrops on the surface of the water. And it's fairly reflective on this flag, but it's light green with black writing and it's 6XH. So this is a great photo. These birds are not eating clams that are as big as you're seeing in the pictures. Their gizzards can only break down clams like smaller than your thumbnail. Here's a great picture one of my technicians took. You can actually see both the bird in the background and the foreground have a leg flag on, so that's a super cool shot. We recorded both these individuals. This bird is on the run. Its leg flag is actually facing the right direction because they can whip all around. They can be turned inside out. They're inside the leg. And he just caught this bird great. Zooming in, you can read this one is 2XP, and this one's dark green with white alphanumerics. And that's helpful for us to know these dark green ones have been tagged much more recently. So those light green ones were more rare. This one's just beautiful lighting. You can see this bird has a little Lamicola clam in its bill. And you can zoom in and read 5CJ. Here's a great example of why photographers were really helpful. So this is a yellow flag. This means this bird was tagged in Mexico, but the ink's worn off. So originally in the field, try as you might with a spotting scope, it just was impossible to read these flags where the ink's worn off. It just looks blank. But when you have a great photo, this is a really zoomed in shot here. You can read the OHH engraved in this actual flag. So this is really awesome. This is what made those photographs valuable, is this piece of data that wasn't very useful because we couldn't get an individual ID suddenly became something that we did have an ID for and we could capture again through photography. And then not everyone used a camera. We also use digiscopes out there. And those, for those of you that aren't familiar, if you buy a special connector for your iPhone or your Android and the brand, make and model of your spotting scope, you can actually connect your phone, your smartphone, to a spotting scope and take pictures. And sometimes these are even better than using cameras because the wide angle lens just lets in a lot of light. So here's an example of a phone scope image, and this is T3+. And this one was really cool because this bird was tagged in Texas, and potentially it's one that switched flyways. It might be a Rufo red knot coming from the East Coast and now is part of the Pacific Coast flyway. So we're not sure what's going on with that individual. This is only the second time it's ever been recorded anywhere. And this last one's just a pretty picture. It's difficult to see, but this is also a flagged bird in flight. Okay, so what are those exciting results? Today I'm going to share with you 3 main objectives. I'll talk about identifying red knot migratory routes and foraging areas, and we'll do this on 3 scales. I'll tell you about prey availability in Controller Bay. We went out and did transects and sampled these core sediments and then sifted the mud and collected all of these small little polychaete worms and little bivalves and amphipods, put them up in baggies, and then we took them back to the lab and sorted through them to figure out what prey was available. And then for that 3rd objective, I will tell you our most recent estimate for the number of red knots that are using Controller Bay and what that means for the importance of this area to this population. So for that first objective, we identified migratory routes and foraging areas on 3 spatial scales. We use MODIS technology to observe flyway routes along the Pacific coast on a very coarse scale. On a medium scale, we identified areas that were important for foraging throughout the larger Copper River Delta complex. So not just Controller Bay, but the Copper River and some sandy barrier islands out there. We also used MODIS for that. And so the image that you're seeing on this slide is a picture of a MODIS tower. And then on a very fine scale, we had observer data in which we used for figuring out the foraging areas within Controller Bay and if birds had site fidelity to those specific areas across 2 years. So in brief, MODIS towers act as receiving stations that use automated radio telemetry to pick up signals from birds with MODIS tags. These tags are really small, they're called nanotags, and you can see from this infographic, they're not just useful for birds. People are using them on butterflies and dragonflies, and the tags send out these unique radio signals that are individually unique and they're digitally encoded. And so these stationary towers have a shared frequency and they can detect tags from as far away as 15 kilometers. Once that tagged bird is detected by the tower, it generates something called a tag report. And that data is stored in a computer that's connected to the MODIS tower. If you're somewhere where there's cell reception, you can set it up so that that data is shooting back to your office If you're somewhere really remote, it might just sit on that computer and then you physically have to get back out to that remote site and pull that computer off and get your data that way. So as of May 2023, there were more than 1,200 MODIS stations across 31 countries, and we had 4 in Alaska last spring. And that was a big deal for us. MODIS is pretty popular in North America right now, but it's just gaining traction in Alaska. The first one was put up in the Copper River Delta area, and I think there were only 2 the year before, and then now we had 4 this year. So it's definitely gaining momentum. But they're challenging because you have to service them. You can't just leave the towers up in Alaska. The wind and snow and rain and weather that we have means that people have to be able to fly out there and service them and put them up and take them down. I mentioned that best case scenario, these birds can be picked up as far away as 15 kilometers, but that also varies on a few things. It depends on the height of your tower. It depends on your antenna orientation. So most of them don't have omnidirectional antennas. You have to know what migratory corridor to face your antenna at, so it's detecting the appropriate angle. And then it depends on other things like terrain and how high that bird is flying relative to the tower. So using that MODIS technology, let's look at what we found on a very coarse scale for red knots that are migrating from Mexico, where they overwinter, all the way up to Alaska, where they breed. This map is showing you a migratory route for 8 different knots. These were all tagged in northwestern Mexico during the spring of 2023 by one of my collaborators named Julian. And the first 2 birds on the legend here are shown in blue and green. You'll see they didn't actually migrate north, and they spent their entire summer in Mexico. And that was really surprising to me. I didn't really realize that they were doing that, but we think that juveniles do that now, and we have the data to back that up. And then there's 6 that did migrate north, and the 5 in this black box were detected in the Copper River Delta complex in my study area in Alaska. Really of interest here is that we often think of these birds as being coastal, that they just stop along sandy beaches and they forage for bivalves and they just pick their way up the coast, or they make these huge long flights without stopping anywhere. And what we're seeing with this MODIS technology is that actually a lot of individuals are being picked up in these inland areas, and they're stopping over in these places that are high and dry, desert-like conditions in California, but usually picked up in places where there is some sort of water source. It might be a marsh, it might be a saline lake. And so that's pretty interesting information. And then of course, you're probably really curious about this bird that has that gold-colored migratory route. This bird was actually picked up all the way in Wyoming. So it got way off course. It was pretty unique because they started moving further south when they first tracked this individual before migrating north. We know that there were a series of storms during the time that this bird migrated, which could have blown them inland, but they migrated at the same time as the rest of their cohort and other birds that were leaving. So we think that this behavior really suggests a lack of experience. Maybe it was this bird's first time doing the migration and they just got a little bit confused on where to go. The other thing I want to point out here is we see these lines going straight across the Pacific without these birds stopping anywhere in BC really, or in Southeast Alaska. We don't know if that's the case because there aren't MODIS towers in those areas. So it could be that they're making more stops than what this map is showing. So next, moving on to that medium scale objective, I really wanted to look at habitat use across the Copper River Delta as a whole. So Controller Bay is seen on the lower right side. There is another tower that we had put up at Alleganic Slough, which is just kind of a muddy slough that's north of that large Copper River. And then there's one more MODIS tower in the area that's on Little Egg Island. And that's a sandy barrier island that's really well known as being an important roost site for a lot of different shorebird species. And so each of these orange dots represents a MODIS tower. And one of the cool things about this is these MODIS towers, because they all operate in the same frequency, these weren't mine that I put up for my project. These were other people's project MODIS towers, but the data is out there and available for anybody to look at. You can go on modis.org and track your favorite shorebird species and find out where they're going. So here's what we learned with that MODIS technology for the Copper River Delta complex. The table I'm showing you provides information on the 5 birds that we detected. All of them were tagged in the Gulf de Santa Clara, which is kind of the armpit of the Baja Peninsula in Mexico. And they were all tagged on the same day, April 24th, 2023. And here's what we learned from their migratory patterns. Most of these birds, 3 out of the 5, used all 3 of these foraging areas in the Copper River Delta complex, indicating that all of these sites are really important. But across the 3 sites, birds stayed the longest at Controller Bay. And generally, birds did not backtrack. So meaning once they moved further north, they didn't revisit more southern foraging areas. You'll note on this table, there is one bird that moved back and forth between Alleganic and Little Egg, but note that these are basically on the same line of latitude. And then interestingly, the length of stay was shorter than what I expected to see. The mean length of stay across the entire Copper River Delta complex was just 3 days. So redknots on the East Coast, that Rufa subspecies, they'll stage at a particular bay for 2 or 3 weeks at a time. And it's just kind of been known that the Pacific population's a little bit different. They don't tend to stay places as long. They fuel up faster and keep moving. But this is a lot shorter than I thought. So that previous telemetry study had a mean stopover of 3 to 4 days in Controller Bay alone. But our sample size is small, so we can't interpret this to mean that the length of stay is getting shorter over time, but it is pretty interesting to note. Moving on to our fine scale objective, we mapped redknot habitat use in Controller Bay from surveys in 2022 and 2023. In general, we conducted these surveys within 2 to 3 hours of the high tide. And so the surveys in 2022 are represented with orange circles, and those in 2023 are in the green. And the size of the circle is correlated with flock size. So that smallest circle is representing 1 to 100 redknots. The medium is 101 to 499, and the large is 500 to 1,000 redknots. So you can see here that the knots are showing a really high degree of site fidelity across these 2 years. These flocks tended to aggregate in areas where there was freshwater intrusion. So the mouth of the Campbell River, adjacent to the mouth of the Bering River, you can't really see it on Ockley Spit, but that area where all the green and orange circles are kind of overlapping, there was also a lot of freshwater runoff going into the bay. And then interestingly, we also noted that we saw larger flock sizes in 2022 compared to 2023. We're not sure why that is. So our second objective was to determine prey availability in Controller Bay and figure out what was available for these birds to eat. And then follow up with that to say, what is it that they're eating? And so overall, we found that Controller Bay yielded a low benthic diversity, and the species composition was really similar to that of the Copper River Delta, where they've done some other studies. Our samples were comprised of 3 main benthic invertebrates. You can see here there's one main one, almost 90% of them was made up of Limacola baltica, and that is the little pink shell in the picture. Secondarily, they were made up with polychaete worms. We just lumped all of those polychaete worms together because they're really difficult to key out to the species level. And then Maya arenaria, and that's that white shell in the picture. So it wasn't surprising to us that the Limacola baltica, that little pink shell, was the dominant prey item. It was the only thing that we saw redknots eating in the field. Even though we saw a lot of other species of shorebirds foraging on polychaete worms, you'd see them pull out a little worm and lean back and try and get these guys out of their burrows. You could see them pick up spoonworms and carry them away. We even saw sometimes birds picking up shrimp. We had an event where a bunch of shrimp washed ashore, but the redknots really only seem to be foraging on bivalves. The Maya arenaria, that white shell, is actually an invasive species, and it was introduced into the West Coast in the late 1800s. We know it's been reported in the Copper River Delta area. It was somewhat surprising to me that it's been found in Controller Bay, given its remoteness, but as you can see, the numbers are quite low. So to our knowledge, this is really the first time the benthic community of Controller Bay has ever been sampled and assessed, and also the first time that Maya arenaria, this invasive species, has been reported here. So because the dominant benthic bivalve was Limacola baltica, we knew that redknots were foraging on it. We decided to just focus on estimating biomass for this species only. And so the plot I'm showing you is 4 different areas where we conducted sampling, and we analyzed them as both near and offshore samples separately to see if there was any difference, and we found no significant difference among the nearshore and offshore densities. But we did find that for offshore densities, they were significantly greater for one particular area, and that was the Eduardes plot, and that's shown in blue compared to those other sites. This was really surprising to us because this was the part of the bay that was really muddy and didn't have any birds and had a lot of bears. And so we really expected that the birds would be eating in areas where the densities were much greater. And then the range of bivalves per core sample was between 53 and 2,200 bivalves per meter squared. This was a little bit lower than I was expecting. Some other published work further north of my study site showed densities of this same species at 3,000 to 4,000 meters squared. So again, this is our study site in the areas of high red knot concentration. And then this blue box represents that area that had the largest densities of Limicola baltica. So we call this the Eduardes camp. So you can see here, wow, we saw really very few red knots. It's pretty surprising to me that they were an area of high density for this bivalve. But there's a few explanations for that. It's possible that the biomass there was higher than other sites because foraging hadn't occurred. So this area still had a lot of prey. It wasn't picked over. It could also be that the higher densities of birds were present and foraging here during the low tide when we weren't out doing the surveys. So maybe birds were eating here just at different times. Or there could be some physical factors at play, the sediment type and salinity. Maybe they really like this muddy sediment in the center of the bay. But in general, like all this information was collected during our pilot season in 2022. So here are the other areas that we sampled, and we just randomly selected them because we didn't know yet where these birds would be foraging. We didn't have this map. And so if you look individually, each of these plots, we kind of just missed the hot spots. So really in that Bering River camp area, we're just on the corner of where birds were foraging. We pretty much completely missed between the Campbell and the Bering. And then even Ockley Spit, if we had just shifted to the left a little bit, we would have been in an area where birds were foraging a lot more. So this could explain why we didn't see those higher densities of 3,000 to 4,000 clams per meter squared like I expected. And in 2023, we really changed this around and I decided to do the exact opposite. And instead of doing that random sampling, I targeted areas where there was a lot of shorebird activity to find out if we had more clams in higher densities. So I don't have that data to share yet, but I'm working on it. So now that we know what's out there, what prey is available, we're interested in understanding the prey that red knots are targeting. So one way to really get at this and understand if they're eating soft-bodied prey items is to use eDNA. And so I'm collaborating with some folks at the University of Maine that have been doing this on red knots on the East Coast. And so far, we collected little fecal samples from red knots, and we also submitted all of the different prey items. So we sent them polychaete worms, amphipods, we caught some really small fish, we sent them shrimp, anything that these birds could be eating, but maybe we're just not seeing them eat. And what they did is they used 2 invertebrate primer sets. So basically, this is used for assessing the DNA of invertebrates specifically. And so they took those red knot fecal samples and tried to pull out the DNA of the prey. Unfortunately, the red knot DNA really swamped out the prey DNA. And so right now they're working to develop what's called blocking primers, which reduces the amplification of the red knot DNA. And I just got an email from them today that the blocking primers were successful. So I'm hoping to have some results on that pretty soon. So finally, what did we figure out for abundance estimates, and did we get enough birds to actually have a robust estimate? We photographed 45 individuals and we recorded 9 resights. So resights are when we see a bird more than one time. And yes, we were able to run that model and we estimated about 9,000 red knots are stopping over in Controller Bay just in the spring of 2023. Based on our most recent population estimate, that's about 40% of the Pacific population. So that's a lot of those birds that are really relying on this one particular bay. We were also able to get some more information like length of stay. And so based on the scans and the surveys that our observers do, the model estimates that at 1.8 days. Again, super short, right? Like we were kind of thinking it would be about 3 or 4 days, but it's really on par with what we saw out in the field. It seemed like every 1 to 2 days, it was a complete turnover of birds And that's probably why we didn't get very many resights. We also estimated that there were 98 birds with flags that stopped over in our study area. And the fact that we actually saw and photographed 45 of those makes me feel really proud. We busted our butts in order to get those numbers. But we know this is likely an underestimate. We're only out there for about 3.5 weeks. We probably miss a few early birds. And I know we miss some birds that come late in June. We also don't cover the entire bay. We cover as much as we can. We hit those hot spots, but there's probably more birds than that. And so as we wrap up, I just want to thank all the people. We're all out there because we love the birds, but we all come back because we love the people that are out there that we work with. And so I'd really like to thank my collaborators, Aaron Cooper with the Forest Service, and many of my ADF&G colleagues, Charlotte Westing out there, the technicians, the undergraduate students, the lab rats, and university professors and international programs folks that have spent and weathered time out in Alaska after growing up in Peru. So it was really a fun project to have a lot of different people involved in. And if there's time before we get to questions, I also have a little video. So this will just kind of give you an idea of like what the weather is like out there, what the birds do. Hi, I'm Jenell Larsen, Tempel, and I'm the species biologist in the Threatened and Endangered Interesting Program at the Alaska Department of Michigan. You're finding me in Control in Alaska. It's a remote coastal region in Alaska, and I'm watching a study on red knots, specifically interested in the Pacific or those alarming invasive scientific members red knot because they're a declining shorebird species. And I'm conducting a site-specific study looking at Control Bay. I'm interested in knowing how many birds are stopping here for almost. And then I'll be collecting eagle samples to determine diet. We're catching in a really great day. Our first week here, we had 80-mile-an-hour winds that were flattening and a lot of rain. So we've weathered the storm and we've been rewarded. We saw our first knots yesterday, and today we're conducting our first surveys and actually seeing red knots. So this fall here, we've got 2 red knots present, and it's just the start of migration. There's a lot more to come. Great. If anyone has questions, I'm happy to answer them.
All right. We'll in the chat. Let's see. All right. I asked, so how do you collect fecal sample from red knots? You don't put diapers on them. So how do you go about, I mean, just seeing the birds and you just explain that to me.
Yeah, that's actually a great question. The first time we did it, there's so many birds. There's 23 different species of shorebirds that are migrating at the same time. There are bird tracks and bird poop everywhere, all over the place. And so you go out there and you have to find a pair of red knots. You watch them for a while, you wait for them to take a poop, and then you leave your spotting scope on it. and you need to send your partner in the field to go collect it. And you need radios because it's so windy out there that you can't hear each other when you're just yelling. And so we would get radios, and so one person would go out and they'd be like, oh, 50 more feet. Oh, 2 steps behind you. And you'd be like, there's nothing there. And they'd be like, yeah, it's right in front of your left foot. And so then you'd guide the other person to exactly where that poop was. We had some challenges because our first season, when we sampled the fecal samples and sent them into the lab, we'd get them back and some of them matched other species. So we had to throw those ones out. And then we worked with someone who was like a fecal sampling expert who came out the second year, and he was able to identify the track size of red knots compared to Dunlin, short-billed dowitchers, long-billed dowitchers, black-bellied plovers, ruddy turnstones, black turnstones, and was able to track down individual fecal samples one at a time. So that was really successful for us the second season.
Wow. Let's see.
So I see one from Amy. How do you use your data for conservation of habitat? Are you partnering with conservation projects who can protect this area? So the first step for us is to publish the data. So until it's published in the scientific community, they kind of say it didn't happen. And so we need to be able to demonstrate that we use these robust methods. We got this population estimate. This is an important site for 40% of Pacific red knots during this time frame, and here's what they're eating, and here's the areas that are important. So the hope is that this information will be used to protect the birds, but if development occurs, you know, that's out of my hands. But if the species gets listed in the future, so the Rufa subspecies is listed on the East Coast as threatened under the Endangered Species Act, and it's a federal process. Whenever they list a species as threatened or endangered, they need to establish critical habitat, and that habitat gets protected. If that should happen to this species on the Pacific coast and this population doesn't recover and they become listed as threatened or endangered, this is really great information that we can share with them to say, hey, Controller Bay is critical habitat. So that's one way that it definitely can have an implication for conservation efforts. This particular area, so I don't know if you're familiar with WISRN sites. It's like the Western Hemisphere Shorebird Reserve Network, are established sites for protection for shorebirds. The Copper River Delta area is a WISRN site, but Controller Bay is not. But Controller Bay is considered an important bird area in Alaska. So it has some level of recognition already. So I hope that answers your question, Amy. Oh, Timothy Lewis, thank you. That's a really nice comment. Jared says, how old are some of these tagged birds? Do you have plans to do any tagging? So that's a great question. The oldest tagged bird on record is 18 years old, or almost 19 years old for a red knot. We know that they live to be in their 20s in the wild. The birds that I've been keeping track of, I actually have been writing down their ages. So once I get those leg flags, I reach out to the bird banding lab, and they're like the federal USGS bird banders, and they submit the permits, and then they get all of that information. And folks like me who are doing research or just curious citizens can reach out and say, hey, I took a photograph of this bird. Here's the photo. Can I get some information on it? And they'll give you a bird banding certificate. And part of that certificate will tell you where and when that bird was tagged. And so that information is cool because then you can say, oh my gosh, this is a 15-year-old bird. And so I've had a few birds that have been like 12, 13, but definitely not the 18, 20-year-olds, not yet. The question of, do I have plans to do any tagging? I would really like to. These are really hard birds to tag. I've been working with a collaborator who I mentioned that's in Mexico. Folks have been trying to tag them in Washington state. They want to redo that population estimate. I said there's about 22,000 red knots. That was published in 2016, but the fieldwork is in 2009 when they did that study. So they want to re-up that, but the tagging hasn't been very successful. And so instead of tagging them in Washington, myself and Washington collaborators have been supporting folks in Mexico to do this. And my collaborator in Mexico says it is extremely hard. Just like Controller Bay, there's a lot of other species, so they kind of have to deal with bycatch issues. And then there's a ton of habitat. Often these are beautiful sandy coastal beaches, but they deal with a lot of disturbance. And so this week, Julian reached out and said, hey, Jenell, it's spring break. I've got to shut down the tagging. There's ATVers and people driving on the beaches and partying, and like, we just can't effectively tag birds in these conditions. So I would love to go down in Mexico and help out on the tagging during the winter season. And that's something I'm working towards. Because we truly only have about 1% of the population tagged on the Pacific coast. The red knots on the East Coast have about 10% of their population tagged. So it's a lot easier to do these mark resite studies when there's more tags in the population. All right, Liz says, Madeline wants to know, what is your favorite bird besides red knots? So that's a fun question. I really like black-capped chickadees. When I was growing up, we had a pair that would nest in our apple tree, and they were so curious. One day I saw one that was drinking water out of the spigot, you know, when you like leave your hose on and there's like water coming out. I went up to him and he just sat there, and I actually pet him with my finger. I like ran my finger down his head and his back, and he let me do that. Just a wild chickadee. And I just was captivated by them as a kid. Do you have a favorite, Madeline?
Chickadees.
You like chickadees? That's cool.
There you go. You inspired somebody already.
Great. Dana says, are there ways others can help you with your research? Combing through video footage, for example, or adding sightings to eBird? Adding sightings to eBird is a great suggestion. I used eBird data after my first year, which was kind of a flop, right? We didn't time migration well. So we were a week early, and then we had to pull camp when it was peak migration. And that was something we just didn't have the flexibility to do. We were kind of booked with these flights with this pilot, and all the field crew, their time was used up and the money was spent for the project budget. So we couldn't stay. But one of the issues with doing these migratory studies is that migrations can change. You know, the peak migration might be May 3rd, it might be the 15th, it might be the 25th. And so I combed through eBird sighting data to try and figure out, okay, when are people seeing red knots in this area? And I was able to do that for various years, and that really helped me establish a better window for my survey. So yeah, definitely if folks are using eBird, like put your data on there. It's super useful. Walt asks, how did these birds get the name red knot? Well, the beautiful pictures that are in this presentation are showing you the knots in their breeding plumage. And so they have that bright red color. But they don't wear that year round. So that's the red part. I don't know why they're called a knot. When I was in the field, we were told that the Spanish name for a red knot is called a fat mud runner. And I think that's actually probably a more appropriate name than a knot, because they start to blow up like little balloons and they just run around the mud flat. So that's kind of fun. Seeing some nice comments, but not any more questions. Anyone else thinks of a question, feel free to just shout it out.
Yeah, Jenell, a lot of other bird families are using the backpacks, the GPS, the cellular backpacks that are put on birds. Is that a candidate for are the red knots candidates for that? I know they can do it on raptors and smaller birds. Or has just the protocol always been kind of these flagging, you get more data back that way?
So I was not a part of any of the tagging efforts that have been done for red knots, but I know for people that try to do these backpack ones, in the past, the birds didn't do very well. So even though they maintained, there's a threshold that they have to maintain for super small amounts of body weight for these birds, it caused some fatalities for red knots, and they stopped using backpack models.
That makes sense then. Okay. I know they were doing some for some of the godwits, weren't it? The bar-tailed godwits that they had set these records for like 2.5 or 3 days. They flew constantly from, was it Alaska to Australia? Is that
Yeah,
correct?
one of my collaborators just gave a talk on this last night, and he was working on the bar-tailed godwit, and they tagged a godwit on the breeding grounds outside of Nome, Alaska. And yes, they used a little harness backpack, like silicone material, and they put a leg flag on it. And a 4-month-old chick left in October and flew all the way to Tasmania. And it was 8,000 miles and I think 11-day voyage for this bird. And this was wild. Like the media went crazy over this because it was the longest ever recorded migration. And yeah, so these backpack transmitters, they can work really well. Unfortunately, it stopped transmitting once that bird made windfall and landed in Tasmania. So nobody knows if B6 is still out there and the backpack fell off, or it got predated, or what happened. So yeah, shorebirds are pretty incredible. My mom asks about drones. I think drones would not be very effective for shorebirds because they have a lot of aerial predators. So in the bay that I'm in, one of the things I added to the survey protocol was to record disturbance. And our first year, we saw peregrine falcons, merlins, a lot of Jaegers causing disturbance. There weren't many eagles because the tree line is really far away, but there's osprey in that area. We had harrier hawks. And then they would even get spooked from things like Caspian terns or Aleutian terns. So even though the terns weren't predating on them, just seeing a large predator flying overhead really spooks the birds. So for this species, I would not recommend drone surveys. Thanks, Shirley and Michaela. Thanks for coming. Nice comment from Scott. Amy says, where are the invasive mollusks coming from? Can they sustain the birds? So that's actually a really good question. These invasive mollusks may have been transported from Vikings. We know that they are an Atlantic species. We know that they arrived in California in the 1800s from oyster farms and have just transferred up probably through spat in the ocean, not so much like ballast water like some other invasives. So we know they're in Alaska, but I also, Amy, like you, kind of think about, well, maybe this isn't a bad thing. Maybe the birds are eating these and it's considered a good food source for them. So that's one of the questions that I hope the eDNA work will answer is, hey, are they eating these invasive species? Karen says, are there any predators that stand out for these birds? I think it was interesting. We saw a lot of predators in 2022, but in 2023, like hardly any predators. The ones that seemed to be bothering them the most were peregrine falcons in one part of the bay. And then in another part of the bay, we had a lot of Jaegers, like Jaegers were even breeding in that area. And we're definitely targeting shorebirds. So I would say those are the top two, at least in my study area.
So how did you time your study, and how long did you um predict, or was it, just money, we only have money, or 2 weeks or 3 weeks, or how long were you out there? Um
Yeah, we are out there for about 3 to 3.5 weeks. We try to be flexible so that we can extend if we need to. I often put in time and tell my technicians they're going to be out there until Memorial Day. And then if the birds are gone on the 25th or 6th or 7th, we pull camp early. I was told by folks, so this is a shorebird mecca for anyone who wants to go see a really cool shorebird festival. The Copper River Delta Shorebird Festival should be on your list. It's based in Cordova. You'll see hopefully some red knots, but also a lot of other shorebird species. It's family friendly. It's super fun. There's multiple festivals throughout Alaska, but this one is particularly timed around the migration. And so the folks I was working with out of Cordova knew this because they planned their festival around it. And they have all these birding field trips, and they go out on airboats on the sloughs. They'll take you out on fishing boats to one of the sandy barrier islands. And so I kind of already had some data from those folks, and that's like citizen science data. So yeah, go to those shorebird festivals, you're helping out scientists. But then also there was some boots on the ground effort from folks that work for the Forest Service there, and they had sent out some of their biologists to go camp out on the sandy barrier islands, to go spend a few nights in Controller Bay and see what's out there. So that was really just developed through those different things. Yeah, I think that kind of answers the duration. Yeah, I used both eBird data, and then there's another person doing shorebird research who has a lot more of experience than I do, and his name's Jim Johnson, and he's doing a breeding study up in Nome, and he also has put tags on birds. And so he shared that information, but the locational data is really rough. So it gives you some idea of where the birds are, but I would go and I would plot these GPS locations with date, and they would end up being like way interior. They'd be like in the Wrangell Mountains, or they'd be like way out in the middle of the Pacific. But I kind of knew like the line of latitude and longitude like made sense that they would be in the Controller Bay area. So I kind of used all those things. I used eBird, I used word of mouth, former studies by Forest Service, and then the small amount of tagged bird information that was available to me. So a question on the chat is, would decoys help you get closer? That's a really good idea. A lot of folks use decoys for tagging purposes on the breeding grounds, but I've never considered using them on the migratory grounds. That might be something to try. That would be a good idea. Do you have any decoys? Did you have any bears cause problems in your camps? That was something I was really proactive about, because once a bear gets into your camp and realizes there's food there, it's pretty much game over, either for you or the bear or everybody. Meaning that we might have to pull the camp. We don't want to have to shoot a bear, but if it's going to be charging into camp, that's, you know, something we're worried about. So anytime in the mud flats that some of those bears came out, if they would get within 300 meters, like we were getting loud, we used marine flares, which are really effective. We didn't have to do any real hazing. You know, we fired off shots a few times just to get their attention and scare them away, but we didn't have any problems with them getting into camp, which was great. We also had bear fences. One of the really cool things that we noticed was that there's a lot of moose in this area, but you never see them. You'd wake up and there'd be fresh moose tracks. And it's like, wow, that's weird. They're just transiting at night. And in the morning, I could actually smell like a moose had bedded down next to our cook tent. And so these females, the Forest Service folks that spend a lot of time doing moose research, will tell us that they'll actually go out onto that long spit, that peninsula there, to get away from bears to give birth. And so they'll go out there, and the pregnant ones will drop calves. And so it was really cool. We actually, one year we saw a pregnant female walk right in front of camp. And that was really fun. But it seemed like they would kind of use us as a buffer. They would know that the humans were there and the bears wouldn't mess with us, and they would come and sleep by our tents. We did have some coyotes get into camp. We actually had some pesky coyotes that would scratch on the tents, and that happened once or twice. Is plumage color based on food source, like flamingos? No, we really don't think so for red knots. That's an interesting question. The little macomas that I pointed out that are pink, they can actually be white or kind of gray or yellow. We think that they are eating fish eggs, little grunion eggs in Mexico, but we don't have anything to really suggest that their color is based on their food. Jeff says, what did you eat while you're out there? We ate a lot of stuff. It was not all freeze-dried meals. We did a bunch of like communal dinners. Our pilot came and dropped us off weekly resupplies. So every week we get fresh groceries. And that was kind of a battle because Cordova is a little fishing town. And so people from all over Alaska that are commercial fishermen show up in May. And so the stores will literally be like cleared out. So before we even go to Cordova, I stop in Anchorage, which is our big city, and then I do a big shop for like freeze-dried food. And we would do like stir fry. We would do, you know, eggs. Sometimes we could do burgers. We'd get some beef. We just got creative. We did do some freeze-dried stuff on the go, but for the most part, we tried to get some fresh veggies mixed in there too. And then it's kind of fun. A lot of the local folks from Cordova really love eating wild food. So some of the Forest Service folks that live there would bring out moose in camp. I had someone fly out dry fish. So they basically had went dip netting for Yulicon. They gutted them and then they smoked and dried them. And we ate those over the fire and they were delicious. What
I
else,
can
you
see,
guys?
I can see this, that is not for the faint of heart.
No, there's really kind of two perspectives that I hear from people in the field. Either they say, I can't believe I get paid to do this, or they just don't pay us enough to do this. And so we definitely want people that are in the former camp. But for the folks that are out there, I mean, it is a really cool environment. Like I've been to a lot of different other places in Alaska, and the outer coast, or the Lost Coast as they call it, is just really spectacular. And yeah, the shower situation, I mean, you're roughing it. We're sleeping in tents. There's no cabin. There's no running water. Like you're digging a hole to go to the bathroom. You can carry the shotgun with you if you want to be in the woods by yourself. You're going to be bathing in a saltwater, freshwater estuary stream while you're out there. But we have a little heated tent. You know, we try to make it cozy and cook together and eat together and have some good camaraderie.
How often did you change camps? Was
Never.
there never? So you were,
We could not easily do that. So the intent with the Zodiac we had at the Bering River camp was that, oh, they could just zip across the bay. It didn't look that far, but it was so shallow, it would take an entire day for them to get there. And then if there was any weather, it just wasn't safe for them to do. So it was like, okay, we'd probably lose 2 survey days if we wanted to switch crews. And we would need to do it at a really good weather window. So it was really difficult to do. We were able to meet up. So that first season, when I was in a camp that was in the middle of the bay with all the bears, none of the birds, we met up with folks that were camping at the Bering River. And so we did this really long day where we each did, I don't know, probably 17 miles, and we like met in the middle and had lunch together. And that was fun. But we didn't really swap out people unless the helicopter or the beaver came out and moved gear and people.
any other questions? Your last chance. What an outstanding presentation, and just kind of show how many folks have been to Alaska.
Well, I
Okay, so
know
we're,
who these people have.
that's what's so wonderful and great about Alaska. It has so many different ecosystems and areas, and they're just huge. Um, been interior. Everybody thinks of, Denali as what Alaska's like, but it has, the islands and the coast and interior and the rainforests and all of that, just makes it that spectacular.
Yeah, it's super diverse. There's a lot of cool places to go. For folks that are really into birding, Nome, Alaska, which is northern and interior. Well, it's on the sound. It's very tundra-y. It's not like southeast. That's a great place to go birding. It really blows up in June. People go crazy over seeing Aleutian terns in that area. In addition to the Copper River Delta Shorebird Festival, there's one in Homer, Alaska on Ketchimak Bay. I believe there's a tern festival out of Yakutat. I don't know if the Stikine River does one too, but yeah, there's some really fun places to come visit and go birding for folks that are interested in making a trip and haven't done that before.
There's a couple new questions there in the chat.
How's this season's research shaping up? It's going good. I'm waiting to get some information back about the eDNA stuff, which I should have within a month. I'm working on new biomass estimates from the benthic bivalves, so that's going pretty good. I've got kind of a mix of a couple people returning, but a new crew this year, so that's exciting. I'm happy to have some folks coming back, but also some fresh new blood. People that are energized. That's another thing out there is like, it's really physical work. Like it's not just, oh, you're kind of roughing it and it's camping and you're dirty. It's just back-to-back survey days, walking in waders on a mud flat or biking in waders, carrying a heavy spotting scope. And so that also can be draining for folks. Scott Ramsey wants to know where their nesting ground is. So the best place to access them will be Nome, off of the road system. Everywhere else that they nest in the tundra, I showed a map and really it just shows you kind of the North Slope and Western Alaska. They're really not very dense nesters. They're territorial. The males get up there first and they kind of defend a territory and they fight other birds off. So I would say don't go somewhere expecting to see this high density of nests. You'd be really lucky to stumble upon one. They're super camouflaged. But if you're trying to go to their nesting grounds, Nome, Alaska would be the best place. Lots of thank yous. Did you make it to Keeluk River? I know it's in the area and I fished it 25 years ago. That's from my Uncle Walt. No, I know that there are about 6 different fishing and hunting outfitters between where we are in our camp and the Yakutat area. So even though we're in the middle of nowhere, there's actually kind of a lot of air traffic because folks are putting in a lot of fishing and bear camps. And I actually did speak to one person who was running one of these camps, and maybe it was that one. It was really known for being like a coho fishing hotspot. And then they also offered some bear hunting. And they asked if, I wanted to use their camp resources for my surveys. And although it would have been great to stay at a nice cushy place where we had a cook and a bunkhouse, it just was not close enough to the bay. So I did not make it out there. Is the nest like that of a killdeer? Probably different. So on the tundra, the birds like to create, they just call it a scrape. It's just like a little shallow bowl. And they'll line it with leaves. They'll go and take like willow leaves, or they'll pick lichen off the tundra and line it. So it's really specific to the habitat that they're rearing their young in. And I will say both the male and the female incubate the eggs together, which is not always common of other shorebird species. So it's kind of cool.
So seeing your pictures there and having wore a lot of waders in my day and done a little bit of biking and optics and cameras and mud, none of that really goes together well. there, how much equipment did you lose, or how do you protect the equipment? That has just got to be a nightmare.
Yeah, that's such a good question. Nothing was really lost yet, but I had some volunteers come out, and these were retired like fish and game and forest service folks. So men in their like 60s, one was even 70, super experienced field biologists for like state and federal agencies. And they told me after this was a long time after the field season. We met up and we were just talking about like results. And one of them goes, "Yeah, I don't really know if we told you, but one time when we were biking, Dan had the gun strapped kind of side saddle on his bike, and we realized later in the day that it had fallen off somewhere in the bay, but we were able to go back and recover it." And that was just like, how do you miss, like, how do you not notice when your huge shotgun like falls off your bike? But they were able to recover it, and I was none the wiser about it, and they cleaned it up and it was fine. As far as optics, one camera got dropped in the muck and the mud, but it totally survived. I usually buy them secondhand, and I buy them from a company called KEH, where you can get cameras and lenses. You can buy them at like a discounted price because they've been used, but they tell you what condition it is. So it's fair, good, excellent. And then you can even buy a warranty for them. So I always try and get like a 2 or 3 year warranty. So it's good for the life of the project that if someone drops the camera or breaks the lens, then we can get it repaired for the next season. So it's definitely difficult. The hardest thing is the mud. Like you want your hands to be dry, so you don't want to be taking your gloves off. You're out there wearing rubber gloves. But then every time you touch something, it gets full of this silty mud, and it gets in your binoculars and your spotting scopes. And so that stuff I send in to get professionally cleaned pretty much every year, especially the tripods. Like the tripod heads will get gritty and they'll just kind of like stop having that nice smooth rotation. So it's definitely hard on the gear. And the bikes don't have a very long life. Some of them will be seeing their last season. Like they've only been out there for 3 years max. Thank you for all the nice comments.
I did see you were using Vortex spotting scopes. At least the covers were Vortex. Is that your preferred optics? Good Wisconsin company.
Yeah, great Wisconsin company, excellent customer sales. They will clean your stuff for free. They'll replace it. I've had them replace tripods for me already. They've always cleaned up the spotting scopes. I love them because of that. Pretty much guaranteed, if something goes wrong, they'll get you a new one, and that's been super awesome. So I would highly recommend Vortex. We use their binoculars too.
So with that, Jenell, I want to thank you so much. Just a fascinating project. And I think we've had more Larsens join our our uh, meeting than ever before. I think we had a new world record on Larsen. So I want to thank all your family for that. Um, so
It's been
uh,
fun. Thanks for having me, Jeff. And thanks to everyone for showing up tonight. I know you guys often do in-person meetings, but I was really happy to see everybody on Zoom.
yeah, so it worked out. Glad to have you. And if you know any of your colleagues or are you again in the future would have anything that you would like to present, we would be happy to have you.
Sounds great. I'll definitely spread the word.
All right. So with that, we're gonna say goodbye. Good evening or afternoon for you up there. So
Bye, everyone. Have a good night.